Good day everyone, I will explain about introduction. For this experiment, we study how environmental factors influence microbial growth. Microorganisms need suitable conditions such as correct temperature, pH, osmotic pressure, and oxygen to grow properly. If these conditions are not suitable, their growth can slow down or stop completely. In the food industry, controlling these factors is very important because it helps prevent food spoilage and foodborne disease. For example, high sugar or salt concentration can inhibit microbial growth by reducing water availability. In addition, some microorganisms need oxygen to grow, while others can survive without oxygen. Therefore, the objective of this experiment was to study the effect of temperature, pH, osmotic pressure, and oxygen on the growth of selected bacteria.
Now, I will present the result of our experiment on the effect of environmental factors on microbial growth. The first one is effect of temperature. At 5 degrees Celsius, there is no visible growth was detected. At 25 degrees Celsius to 37 degrees Celsius, there is poor growth was observed. However, at 50 degrees Celsius, the growth decreased significantly. Therefore, at both 25 degrees Celsius and 37 degrees Celsius, poor growth was recorded, suggesting that the microorganisms do not grow efficiently at these temperatures. The second one is effect of osmotic pressure, salt. For salt concentration, high growth was observed at 0%, while at 1% of salt shows poor growth rate. As the concentration increased to 5%, the growth began to decline. At 10% of salt concentration, no growth was observed. This suggesting that the increasing salt concentration inhibits microbial growth. The third one is effect of osmotic pressure, sugar. For sugar concentration, no growth was observed at 0%. At 1% and 10% of sugar concentration, moderate growth was recorded. At 20%, good growth was observed. This indicates that in this experiment, high sugar concentration appears to support microbial growth. The fourth one is effect of pH. In terms of pH levels, no growth was observed at pH 3. At pH 5 and pH 7, moderate growth was recorded. At pH 10, good growth was observed. This result suggests that microorganisms in this experiment grow better in alkaline conditions. The last one is effect of oxygen. For oxygen conditions, moderate growth was observed in the presence of paraffin oil or anaerobic condition. Meanwhile, good growth was observed without paraffin oil or aerobic condition. This indicates that the microorganisms grow better in the presence of oxygen, although they are still able to grow without it.
experiment, we know that the microorganism used was a pure culture of Escherichia coli grown in tritic soy broth, TSB. The growth of E. coli was tested under different environmental factors including temperature, osmotic pressure, which is salt and sugar, pH, and oxygen availability. The results show that these factors significantly influence bacterial growth. The first one is effect of temperature on growth of E. coli. Based on the observation, E. coli showed better growth at 25 and 37 degrees Celsius, while lower growth was observed at 5 and 50 degrees Celsius. The broth at 37 degrees Celsius appeared more turbid, indicating higher bacterial growth. This is because E. coli is a mesophilic bacterium that grows best at moderate temperature. Low temperature slows enzyme activity and metabolism, while high temperature may denature proteins and damage enzyme required for growth. This result is supported by Madigan in 2021, who reported that E. coli has an optimal growth at temperature around 37 degrees Celsius. Prescott also stated that refrigeration temperature slows the multiplication of E. coli. The second one is effect of osmotic pressure on growth of E. coli. The results show that growth was highest in 0% and 1% salt, moderate at 5% salt, and lowest at 10% salt. As salt concentration increased, bacterial growth decreased. This happened because high salt concentration creates a hypertonic condition, causing water to leave bacterial cell through osmosis. Loss of water leads to plasmolysis and reduced cell activity. According to J in 2018, E. coli is not salt tolerant and high sodium chloride concentration can inhibit its growth. Therefore, the experimental result is consistent with the literature.
Based on the result, growth was higher in lower sugar concentration and reduced at 10% and 20% sugar. This indicates that concentrated sugar solution inhibit bacterial growth. High sugar concentration also lowers water activity and cause of osmotic stress, where microorganisms lose water from the cell. This result in agreement with Bouchet 1981, who reported that sugar preservation such as jam and syrup inhibit microbial growth because of reduced available water. Effect of pH. The observation showed that bacterial growth was poor at pH 3, better at pH 5, and highest around pH 7, while growth decreased again at pH 10. This indicates that the test bacteria prefer neutral pH condition, very acidic and alkaline. Environment interferes with enzyme and structure, membrane transport, and nutrient uptake. This result is similar to finding by Tortora et al. 2020, who reported that most bacteria grow best at pH 6.5 until 7.5. Acidic condition below pH 4 usually inhibit many pathogenic bacteria. Effect of oxygen. From the result, growth occurred both with paraffin oil layer and without paraffin oil layer, but growth was higher without oil. This suggests the test bacteria may be facultative anaerobe, meaning they can grow in both presence and absence of oxygen, but better in oxygenated condition. Paraffin oil limit oxygen diffusion into the medium, creating anaerobic condition. This finding agrees with Madigan et al. 2021, who state that facultative anaerobe bacteria such as E. coli can use oxygen when available but can switch to fermentation anaerobic respiration when oxygen is absent.
Lastly, I will explain about conclusion. In conclusion, microbial growth depends greatly on surrounding environmental conditions such as temperature, pH, osmotic pressure, and oxygen availability. These factors directly affect the availability of microorganisms to survive and reproduce. Different bacteria respond differently to these conditions based on their physiological characteristics. For example, some bacteria grow well in the presence of oxygen, while others grow better without oxygen. Some can tolerate acidic conditions, while others require a natural pH. Optimum environmental conditions promote faster microbial growth, while unsuitable conditions can slow down growth or completely inhibit microorganisms. This is supported by previous study report by Welter et al. 2024. Therefore, proper control of environmental factor is very important in food processing and storage. It helps prevent contamination, reduce spoilage, extend shelf life, and ensure food safety for consumer. That's all for me. Thank you.